Yeah, okay. Yeah. Hi, good afternoon, good morning, all. And today, as Elizabeth said, we have two talks, and the first talk will be by Dr. Anvita Kerkar, and who was a PhD student at the NCPOR, National Center for Polar and Ocean Research. And after her PhD, and she is presently working at Portland University in the US. And she has been working in the Indian sector of Southern Ocean on the topic of phytoplankton production and biooptics. And today's talk for Nivitas will be the work she has carried out during her PhD degree. So with this short introduction, I would like to invite Anvita to give her talk. Anvita, the floor is yours.
Thank you. Is my screen visible?
Yep, that's perfect.
Great. Yeah, so thank you for the introduction, and thanks for having me here today. I would be presenting my talk on the primary productivity in the Indian sector of the Southern Ocean, and I would be discussing my observations from 3 Austral summers. So just before I begin, I would like to give a brief introduction of my present work. I'm working as a postdoctoral researcher at the Department of Biology, Portland State University in Oregon. And currently, I'm studying the selective feeding responses between the gelatinous grazers and picophytoplankton. And I'm using molecular methods like qPCR to quantify these interactions. Prior to this, I was working as a postdoc at Florida Atlantic University, and I was studying the applications of digital holography in plankton ecology. And prior to this, I was a student, PhD student with Dr. Sharath Tripathi at National Center for Polar and Ocean Research. That brings me to my present topic on phytoplankton productivity and biooptical variability in the Southern Ocean and coastal Antarctic region. I have been a part of 2 scientific expeditions to the Southern Ocean, and I would be discussing the observations which have been a part of my PhD thesis. So a brief outline of my presentation is I would be starting with a brief introduction to the Southern Ocean primary productivity and its potential controlling variables, and my observations on the measurements of primary productivity and an intercomparison of the primary productivity estimates from different methods. I would be very briefly going through the potential modulators, focusing on the biooptical variables and grazers or the copepods in the region. So a few weeks ago, I came across this interesting model that was developed by MIT that shows the global distribution of phytoplankton. And it was very interesting to see these diverse and teeny tiny cells which have a great impact on the globe, which by accounting around 50% to the global photosynthesis, these tiny cells are so responsive to the surrounding physiological and biological variables, and these give invaluable insights in the changing oceans. So by this, as all of us are aware today, the climate change is a triple threat for the oceans, making them warmer, less oxygenated, and more acidic. So several impacts such as sea level rise, bleaching of calcareous organisms, corals, recurring toxic algal blooms, habitat loss for several species in several aquatic ecosystems, acidified water, and economic disruption of fisheries and several other sectors is becoming more and more evident over the years. And Southern Ocean plays a critical role in this scenario. So the Southern Ocean is characterized by several unique properties such as oceanic frontal structure, the contribution to total global primary production that's remarkable. It hosts large stocks of zooplankton and has a profound influence on the climatic system. It's also regarded as the high nutrient, low chlorophyll region, and it's an unused repository of macronutrients. Southern Ocean forms a critical component of the carbon cycle, and it's a carbon dioxide sink. It stores huge quantities of heat and carbon dioxide, and also connects 3 oceans, that's the Indian, Atlantic, and Pacific Oceans. And the topic of my today's discussion is its contribution to global primary production. So coming to the fundamentals of the process, primary productivity can be defined as building up of organic matter from inorganic elements. And this process is mediated by primary producers, which are right at the base of the marine food web. So the rate at which primary production occurs is defined as primary productivity. And these primary producers occupy the base of the trophic pyramid. And as we can see, the trophic pyramid is fueled right by the primary producers. So therefore, primary production is the source of the all organic biomass that leads to the carbon production and export over the entire food chain. Primary productivity is controlled by several variables. Some of the major ones are light. The water column is basically constituted of phytoplankton and non-phytoplankton components, and the optically active properties of these constituents are assessed to study the impact of optical field on the primary production. The second one is the nutrients. The macronutrients such as nitrates, phosphates, silicates, and the other micronutrients have a profound influence on the biomass and community type of the phytoplankton in every ecosystem. Another one is grazing by the predators, and the immediate level of primary consumers that can be represented by some abundant class like copepods or gelatinous grazers are the immediate ones which clear the phytoplankton community. And another controlling variable is the phytoplankton physiology or the health of the phytoplankton community. So some of the variables such as fluorescence or the photochemical yield are proxies for the physiology of phytoplankton. So in my study, I measured the phytoplankton productivity variability and made an intercomparison of the productivity estimates using more than one methods. So basically, the sampling was carried out in the Indian sector of the Southern Ocean during 3 austral summers. And this was the transect of sampling, and the annotations indicate the austral summer of sampling. So the productivity was measured through incubations in a deck tank, and the controls were, of course, filtered immediately, and the samples were packed in appropriate light filters to simulate the oceanic depth from which they were collected. Oceanic PP has been measured through several methods and approaches to date, and it could be evolution of oxygen or incorporation of isotopes or active fluorometric methods or the satellite-based methods. But all of these methods have a basic aim of improving the estimation of primary production at the global scale. So I used 3 of these methods. Firstly, it was the 14C or the radioactive carbon-based isotope method, which measured the disintegration per minute or the DPM values, which were converted into the daily rates of primary production. Secondly, it was the stable isotope method, which converted the delta 13C values to the rates of primary production. I'm sorry. And the next one was an instantaneous method to measure primary production, which through this equation calculated the instantaneous rates of primary production. And along with it, it also measured the physiology of the phytoplankton community. So the proxies were derived through the ratios between the variable and the maximum fluorescence and some similar parameters which gave an indication about the phytoplankton physiology at the time of sampling. So some of the factors which were evaluated were the variability of chlorophyll A and primary productivity across the water columns. So this showed a significant correlation at several stations and several depths. So in general, the chlorophyll A and primary productivity showed a north to south increasing concentration gradient at most of the stations. And there was a presence of photoinhibition effect, which was assessed through PAR-PB relationship for the community. And in general, the correlation between PP and chlorophyll A was significant over the region. During the second austral summer, the frontal stations were evaluated for biooptical variability as well as primary productivity. And some of the key findings were the frontal stations being consistently less productive, And this could be attributed to lower estimates of chlorophyll A, and also the silicates were low in this region. There was also a package effect, or the pigment packaging effect, that was prevalent at the frontal stations. So these all together could be linking to the differences or variability in primary productivity during these austral summers. There was another interesting factor that was measured by comparing the PP estimates from different techniques during the third austral summer. So the primary productivity was measured using fast repetition rate fluorometry, and these rates showed a strong correlation with the radioactive based method measurement of primary production, as well as the stable carbon isotope method. And these range of estimates was also compared with the literature, which was seen to show an agreement. And there was a low magnitude of discrepancies between the estimates that were obtained. Therefore, one could say that FRRF could be a potentially reliable tool to estimate primary productivity in the Indian sector of the Southern Ocean. However, one should be cautious about the sample size, technical differences, and assumptions that are associated with every technique of primary productivity estimation. The FRRF was also used to evaluate the photosynthetic efficiency and the other photophysiological parameters of phytoplankton. So both the variable and maximum fluorescence ratios were shown to decrease exponentially or across the water column. So this is an indication of general low photosynthetic efficiency of the phytoplankton community in the region. So the values ranged between 0.1 to 0.4, and this is a typical low photosynthetic efficiency of phytoplankton. And as it is seen, it's a characteristic of Southern Ocean phytoplankton to be acclimatized to low light. So this can be a further validation to the results. To infer, the PP estimates showed a north to south increasing gradient. Photo inhibition was evident through PAR-PB relationship, and further, this could be also linked to the low photosynthetic efficiency of the Southern Ocean phytoplankton. And in case of intercomparability of the PP estimates, the 13C and FRRF PP trend continued to be reliable over the Indian sector of the Southern Ocean. Coming to the biooptical assessment of this region, biooptical oceanography basically relies on the interactions between the light and life in the ocean. So it's measuring the biooptical constituents across the water column. So these are typically the phytoplankton, non-phytoplankton, total suspended matter, and the chromophoric dissolved matter. So all these are measured to get a picture of the absorption that occurs in the water column. And this can have great implication on the primary productivity of the ocean. So the characteristic absorption properties of each of these optically active substance is measured from the absorption spectra. So here, basically, the absorption of particles was measured using a UV visible spectrophotometer. And further, the samples were treated with methanol and remeasured in order to get the non-phytoplankton component on a filter paper. So when the non-phytoplankton component was subtracted from the total particle component, it led to the absorption by phytoplankton. So this is how the three variables were derived. And parallelly, the acidum, or the chromophoric dissolved organic matter, was also measured using the UV visible spectrophotometer. So coming to spectra from the filter papers, this is just a gist of different absorption spectra during the 3 austral summers, and the peaks at specific wavelengths were further evaluated to get a flavor of the biooptical variability in the region. Bringing the spectra to graphs, the PP was better explained by the absorption by phytoplankton compared to the biomass or the chlorophyll. The primary productivity and APH at 443 nanometer ratio declined as we progressed to the deep chlorophyll maximum from the surface. And the pigment packaging effect was prominent at the frontal and coastal domains. So definitely it was something constant and modulating the primary productivity throughout the Indian sector of the Southern Ocean. These interactions were evaluated over all the austral summers. They were pretty constant, and the A star pH and chlorophyll relationship indicated pigment packaging effect at discrete depths, starting from the surface, also at the DCM, and also in between. And the pigment packaging effect was also validated further through the B/R ratios. Let me come to that. So the phytoplankton community was characterized. So basically the validation was kind of a two-step validation. Firstly, it was the B/R ratio, and it's the ratio that's used to classify the dominant phytoplankton functional groups or classes based on their size. So this was the ratio between the absorption of phytoplankton between 440 and 675 nanometers, which divided the phytoplankton community into pico, micro, and nano phytoplankton. This was also validated by applying the data to an absorption-based model, which was used to classify the dominant phytoplankton sizes. Here, the slope of APH at 443 was something that was examined to get a picture of the phytoplankton community. The observations were constant, and the phytoplankton size structure showed that the most dominant community type was of microphytoplankton, followed by nanophytoplankton, and the least were picophytoplankton. So this was constant over both the austral summers. And however, there was an observation that the model in general captured microphytoplankton, or the largest sized phytoplankton, lesser in case of both the tests. So to infer, most of the stations were dominated by larger phytoplankton type, and that could be a reason why the pigment packaging effect was more prevalent across the Indian sector of the Southern Ocean. Also, this was validated through the negative relationship between the A star pH and chlorophyll A, which again corresponds to package effect. And the absorption by phytoplankton was a better predictor of primary productivity in the surface waters in the region. Also, the global absorption model was seen to capture the smaller sized phytoplankton. However, it was not as efficient in evaluating the larger phytoplankton or microphytoplankton. Therefore, this could be one important recommendation or highlight which recommends necessity of region-specific modifications in case of Indian sector of the Southern Ocean. And this would have implications for future model-based primary productivity studies. So these observations are a start point for region-specific ocean color models or ecological models, which would be aiming to derive the phytoplankton size structure or maybe physiological effects of phytoplankton, physiological status of phytoplankton in the region. I will quickly run through this. This was a very initial attempt to evaluate the trophic link between the primary and secondary producers through a theoretical carbon-based approach. So mesocosm or grazing studies were something which were not conducted during any of these experiments. This was just to check the first step in energy flow from phytoplankton to the zooplankton community in the Indian sector of the Southern Ocean. And some of the highlights of, let me come to the methods first. The estimation of carbon biomass of the copepod community was done through morphometric equations. The specimens were counted in Bogorov's chamber and identified using some authentic taxonomic keys. And the carbon was further estimated using the imaging software and established chromosome length measurement based equations. And also the equations that helped in measuring trophic link between the primary and secondary producers was applied to the data sets. Species-specific carbon content could be evaluated using these equations, and this was done for 2 austral summers. So these are in general the contributions of each of the species And this can, of course, at a cumulative level be extrapolated to the class level carbon stocks or even community level carbon stocks. And these studies are extremely scant in the Indian sector of the Southern Ocean. Further, there was a strong correlation between the carbon stock of phytoplankton and the zooplankton, and there was a clear variation in trophic efficiencies seen between these two communities. So it indicates as an important method to evaluate the effective transfer of energy between the producers and the zooplankton in the community. Another highlight of the technique was it allowed the sample preservation. So the samples are still in the laboratory and can be utilized for anything else or any different kind of experiments. So it also told the amount of carbon available for the next trophic level and amount of carbon that was contributed by each species of copepod and the entire copepod community during the study. So the species-specific carbon estimates in general are extremely sparse at a global level. And these are very essential in predicting the prey-predator interactions and several other ecological interactions at regional as well as temporal levels. So such estimates could help a long-term monitoring and clearance rates and changing climate or changing water conditions, and definitely improvise the picture of the food web in the aquatic ecosystems. So these are also a start point, I would say, to the studies, because right now the ecological models do not account for such interactions, especially when it comes to small phytoplankton. So these studies, if tweaked to smaller phytoplankton communities, can definitely be very helpful. So to summarize the study, the estimates of FRRF were closer to the stable isotope-based primary production. So this could be a potentially promising technique to estimate primary production at a relatively higher resolution in the Indian sector of the Southern Ocean. Further, it could also be used in standardizing the stable carbon isotope technique, which is a non-invasive way to estimate primary production. And it could also have an impact over the current measurements, which include radioactive contamination, and potentially solve the problem of sparse PP estimates in the Indian sector of the Southern Ocean. FRRF, along with the primary productivity estimation, can also help in getting a better picture of the photochemistry and the physiology of the Southern Ocean phytoplankton at an extensive scale. And coming to the biooptical characterization, the results have suggested a necessity in regional modifications for the absorption-based models when applied to the Indian sector of the Southern Ocean, especially emphasizing on the A star pH and chlorophyll A relationships, and the size structure of the phytoplankton that was evaluated from the spectral absorption also has suggested some cautions, especially like while mapping the larger phytoplankton in the Southern Ocean before the models are applied to the global absorption models. And the species and community carbon estimates could be evaluated at from a non-destructive technique. So this could be a start point to monitor the carbon flux at regional levels or specific time points, and get a better picture of the trophic link between the phytoplankton and zooplankton and food web in general. With this, I would like to extend my thanks to SOOS and Southern Ocean Indian Sector Studies. I would like to thank Dr. Sharat Tripathi, who has been my PhD advisor, and Dr. Sabu, Dr. Pandi, and Venkat Ramana for their contribution during my PhD, the entire Ocean Science Group at NCPOR, the funding agencies, captains and crew, and all the collaborators, my family and friends. Thanks for this wonderful opportunity, and thanks for the patient hearing.
Thank you, Anvita, for your nice talk. And may I request Elizabeth, if there is any questions in the chat box, may I kindly read out. or can I take the questions? Elizabeth, can you hear me?
Yeah. Yes,
Yeah.
I can hear you. Yeah, I think if anyone has a question, I don't know that we'll be able to see raised hands, but we can try.
Yeah, that will be fine. If anybody has any questions, please feel free. Okay, Rob, please.
Can you hear me?
Yes.
Yeah.
Okay.
Oh, yes, thank you.
Rob, yeah, please go ahead.
Oh, thank you very much indeed. Hi, Anvita. Thank you very much for a wonderful talk there. That was really interesting. I'm coming from a SOIS perspective, and I was just wondering to what extent SOIS has influenced your findings and measurements and relationships. Are the species that you've been measuring related specifically to SOIS in any way at all, please?
Thanks, Rob. That's an interesting question. And the influence of sea ice in particular was not measured in the study, so I'm not sure how that can be answered. And secondly, the phytoplankton community type or the functional groups were basically coming from the spectral measurements, so that was not a direct measurement. So yeah, that's all I can say.
Yeah, thank you very much, Anvita. I was wondering as well if within the Indian program whether anybody is looking at the sea ice and its influence on primary production.
Right. Yeah, that would be really interesting. Yeah.
Rob,
thank you very much.
Yeah, Rob, at present, I think no one is looking that. We are looking at CIs. There are people working on CIs, of course. But as you raised this question, this is very interesting. Nobody's looking into that, in fact. But we should be doing it soon.
Thank you very much, Sarat. And thank you again, Anvita.
Toru Hirawati, please.
Thank you. Thank you very much for many information on the primary production in this assumption. So you studied about the relationship between the FRF and the carbon 13 or carbon 14 primary production. So in the final slide, you said that you will use FRF to derive the primary production in carbon as a carbon amount. So do you plan to use just only the FRF, or so still continue the carbon-based measurements simultaneously?
Please correct me if I'm wrong. But from what I understood, you asked if I used FRRF to compare the primary productivity through carbon. So that was, I mean, FRRF measures the productivity in an indirect way. So it takes into account the electron transport rate, and that is further converted to the estimates of carbon. And so in this way, it was kind of an extrapolated attempt to get the rate of primary productivity. And this was compared to the carbon-based estimation from the 13C and 14C techniques. And this is still in progress. So I'll be, again, reworking this stuff.
Yes, I think so. And also maybe the phytoplankton group is gradually changing under the climate change. So the relationship between the carbon and the fluorescence information will maybe changed. So I recommend you to continue there. So both FRF and carbon measurements for the primary production.
Okay. Yeah. Thank you very much.
Thank you very much.
Yeah, thank you, Professor Hirawati. Actually, Professor Hirawati was asking, our comparison result is so robust. Do you think that you can only measure FRF and rely on it? So I hope not. We have to still continue with carbon measurements. That was the question. Actually, and,
okay. Yeah, I'm sorry.
A long way to go.
Okay.
Thank you.
Thank you.
So I can't see all the hands, I don't think, but I might just ask you, Anvita, thank you very much for your presentation, to stop sharing your screen, and we can, of course, continue in the chat, but we might now switch to our next speaker.
Thank you, Anita. You can sign out.
Thank you. So next up, we have Annie Foppert, who will, there she is. And so I'm very pleased to introduce Annie. So Annie is a research associate at the University of Tasmania, which is just next door to where I am here in Hobart. And she is, of course, contributing to an Australian program called the Australian Antarctic Program Partnership. And most recently, a nice line that she has added to her CV was her heroic task of being the chief scientist or the voyage leader on a recent voyage to the Southern Ocean, which I believe is going to be the focus of her presentation today. So I will hand it over to you, Annie.
Okay, thank you, Elizabeth, and thank you. My screen's all messed up. Thank you to Elizabeth, Kai, and Sarat for organizing this webinar. It's really great to be able to talk to people about what I and many others have been doing here in Hobart. So, yeah, most of this talk will be kind of about the research plan and strategy and approach, and not so much on the results, although I hope to have time to get to some of the results at the end, mainly because this is about a voyage that we undertook this summer that we've only been back for a little over 2 months now. So results and analyses are underway, but I think it's a really nice framework to think about the Southern Ocean and the atmosphere above it, and how they're all linked and how they affect the Earth's climate. So yes, the voyage was a multidisciplinary investigations of the Southern Ocean, or MISO, and the goal was to link the physics, biogeochemistry, plankton, aerosols, clouds, and climate. So no menial task here. As Elizabeth said, I was the chief scientist on board. Steve was chief scientist providing shore support, and there was a whole team of PIs and people on board and on shore that helped make this voyage happen. I am a physical oceanographer at the University of Tasmania and Australian Antarctic Program Partnership, so I'm not an expert on all of the biogeochemistry, plankton, aerosols, clouds, and climate part of it, but I hope that I'm able to articulate the importance of all those things as well. I guess our mission statement is could be thought of to be the goal of MISO is to improve understanding of how the Southern Ocean influences the Earth system and use this knowledge to improve climate projections. I'm kind of preaching to the choir here, but I'll go through some background anyway. We know that the Southern Ocean has a profound influence on global climate, biogeochemical cycling, and biological productivity. It takes up anthropogenic heat and carbon dioxide, but that also means that it's going to have a large influence on sea level rise and ocean acidification. And the Southern Ocean is adjacent to the Antarctic ice sheet, and that ice sheet is the largest and importantly the most uncertain contributor to future sea level rise. And so the Southern Ocean plays this crucial role in the Earth system. And yet, Earth system models currently have large and persistent biases over the Southern Ocean. Here is a picture of shortwave cloud radiative bias between the Australian atmospheric model, the ACCESS model, and observations. And you can see that over most of the Southern Ocean, we're not getting the atmosphere right. And it's thought that that's mainly due to we're not getting the right cloud formation and the right types of clouds forming over the Southern Ocean. And so our big picture science questions are, and so our goal is to fix those biases in the climate model so that we can make better projections of future climate. And so the big picture science questions are what physical and biological processes and interactions account for the unique aerosol cloud precipitation radiation interactions over the Southern Ocean. So clouds in the Southern Ocean form a bit differently than they do over the rest of the globe, mainly because there's not many continental sources of dust and aerosols. So it's thought that the biology in the Southern Ocean actually produces precursors, releases gases that then turn into aerosols in the atmosphere that the clouds can form on. So we want to understand that connection, which is kind of that second point there, that what are the controls on the primary productivity and the community compositions, and how do those affect the production of these biogenic aerosols or precursors. We want to know how and why the ocean inventories of heat and carbon are evolving in time. So large scale overturning circulation kind of questions here. And what are the impacts on climate and sea level rise? And a big focus of this project is on the high latitude Southern Ocean, how it's changing and the implications for the stability of the ice sheet, the formation of Antarctic bottom water, and the deep overturning circulation cell, which we know are all very important for the future. And so the fully coupled research questions that I've just presented you require a fully integrated multidisciplinary approach in order to answer them. And so the Southern Ocean is quite special because everything is really linked together. And so, for example, if you have a change in the overturning circulation here, you will change the anthropogenic heat and carbon inventories in the ocean. But not only that, you'll change the iron and nutrient, the macro and micronutrient supply to the surfaced ocean, which in turn affects the biological primary productivity and community compositions at the surface. Those community compositions and phytoplankton communities could impact the different biogenic sources of these cloud condensation nuclei that are released into the atmosphere, mainly dimethyl sulfide, DMS, which form these aerosols for the clouds to form on. Changes in the cloud then influence the Earth's radiative budget, which then affect the amount of heat and going back into the ocean. So everything is linked together, and it's really difficult to understand how the Southern Ocean influences the Earth climate by only looking at one of these aspects at a time. So the picture on the right there shows sort of a bit of a tangled web of how all these things are connected, and that's really what I want you to get from that, is that you'd be hard pressed to find one of those that isn't linked to all of the others, if not 4 of the 5 other bubbles over there. And we need to observe the system to understand it, and we then need to develop and implement new parameterizations to improve the Earth system models. So that's the big goal. And we set out in January to collect the observations to do the first part of that process. And so our research strategy was complicated, but we did 65 days on the RV Investigator, the CSIRO and Australian Marine National Facilities research vessel, starting in Hobart and going to the Antarctic margin region, and then westward along the slope there, and then back up the Go Ship repeat section of I-9 south to Fremantle in Western Australia. Along the way, we had kind of a 5-pronged approach to observations. So we had the underway data that the ship is collecting all the time, including physical, biogeochemical, and biological parameters. We also had atmospheric measurements going pretty much all the time, measuring the cloud precipitation and radiative properties. We were launching radiosondes to understand the meteorology, and we also had onboard sea spray generation tanks to understand how different biological controls and physical properties of the water affected how much sea spray was generated to produce cloud nuclei. We did repeat stations, which included the GOSHIP and GeoTrace's, I guess, standard stations. And so this was the first time that the I-9 or the GS05 is the GeoTrace's name for that line was occupied to do the trace element and isotopes, both dissolved and particulate. So we had both a trace metal clean rosette and in situ pumps that were deployed at multiple depths at many of the stations. And then the hydrography, the CTD LADCP, that is well known. A novel aspect of the MESA voyage was 4 multi-day process stations. So some work has shown that that some of these processes that control the biological communities need more than just a single occupation to understand. So we had roughly 3-day stations. We occupied stations for 3 days, which included deckboard incubations to look at, for example, ocean acidification, ocean alkalinity enhancement experiments. And then we also had 2,000-liter mesocosm incubations on the front deck that we filled with trace metal clean water and infused one of the tanks with iron, and it was totally sealed. And then we were able to sample both the water and the headspace, the air in the headspace above it, to directly quantify the release of these biogenic precursors that were being released in the tanks with and without iron. So you could see how the effect of nutrients were directly influencing the biological release of these precursors that the clouds can form on. We also had 22 autonomous floats that we deployed. We did 10 biogeochemical Argo floats from the SOCOM project in the US and 12 deep Argo floats, 4 deep Arvor floats from France that were also equipped with oxygen sensors, and 8 deep solo floats that Australia provided, the AAPP provided. So that is very exciting as well. And lastly, we used remote sensing to not only choose where these process stations would be, but to help us make decisions along the way. Okay. And so just in the last 5 or 10 minutes now, I'll talk about probably one of these questions that I'm the most comfortable talking about, which is how is the high latitude Southern Ocean changing, and what are the implications for, I'm particularly interested in Antarctic bottom water and the deep overturning circulation. And so just a bit of background on that is that we know that Antarctic bottom water is of global importance. It makes up the lower limb of the overturning circulation. It forms near Antarctica in only 4 locations, despite making up 40% of the global ocean volume. As it sinks into the abyss, it takes heat and carbon with it, storing it for millennial timescales, and it also brings oxygen into the deep ocean. And so the picture here just shows the fraction of Antarctic bottom water in the deepest CTD sample. And unsurprisingly, it's found in the most highest concentrations near Antarctica, but even far north in the Pacific and Indian basins, it's found all the way to the most northern parts of the basin. The North Atlantic, it sort of dwindles away where you have North Atlantic deep water forming. Again, I'm kind of preaching to the choir here, but the Southern Ocean is notoriously harsh, especially in winter, and to measure Antarctic bottom water, you need to get to the bottom of the ocean. And so it's outside of the core Argo capabilities. This picture shows hydrographic profiles reaching deeper than 2,100 meters, so non-core Argo float profiles in particular. And you can see, as we know, the Southern Ocean is a real data desert. And so we rely on these hydrographic sections to track changes in Antarctic bottom water. And I've highlighted there in purple the kind of meso area that we were covering. So from 150 east, repeating the southern end of SR3 and then up I-9. And we rely on these sections to tell us not only how the ocean is changing, but why. And so this is a picture from Van Wyck and Rintoul in 2014 that shows how the ocean, the Antarctic bottom water is changing, with the blue bars representing freshening and the red bars representing warming at a bunch of repeat locations. And the MESO voyage track crosses this pathway in 5 locations. So we have also the along stream evolution of how that bottom water is changing. We know that bottom water especially has especially strong signal of change in the last couple of decades, which is strongest in the Australian Antarctic Basin, which is where we are focused. And we see that that layer has contracted by about 50% since 1970 to the early 2010s. And that's along I-9, which we repeated earlier this year. This is a modeling study that shows, again, this slowdown of the overturning circulation as seen by freshening and warming of the abyssal ocean. You can see those changes are, again, super strong near in the Australian Antarctic Basin, and it's driven by the glacial melt. So the freshening, the freshwater budget on the shelf is what's controlling the production and export of these properties, the bottom water. And so we want to be able to track how that's going in time. And then quickly, I think most people wouldn't be familiar with Deep Argo, but Deep Argo is a fairly new program that has floats that can profile down to 6,000 meters or 4,000 meters, depending on the float type. And so the Australian Deep Argo or Argo program, along with international collaborators, started a pilot array in the Australian Antarctic Basin in 2018, which provides year-round full depth profiles, filling that seasonal bias in the Southern Ocean. And they also safely profile under sea ice. So they go under the sea ice for the winter, they continue to profile, and then as the ice melts, they relay the data back via satellite once they are able to get to the surface again. And just in the first 5.5 years of this pilot array, which started as 12 floats and has been continually reseeded as the years have gone on. So as of April last year, the array has provided over 2.5 times the number of deep profiles and 10 times the number of deep winter profiles in that southern sector of the Australian Antarctic Basin, compared with the World Ocean Database. So this is really changing the way that we understand the variability of the deep ocean on as short timescales as even daily and seasonally, and then filling in the gaps in the multi-decadal time series. And so in order to study bottom water on this voyage, it fit in these 5 sections along the bottom water outflow pathway, which included stations on repeat sections at the southern end of P11 and SR3, the 132 section and 123 that are nearby other historical CTD stations, and the southern end of I-9. And we deployed the 12 deep floats in this southern sector. We also had 3 shelf stations there in the green triangles that were in or very close to the Adélie Depression, which is where Adélie land bottom water, one of the 2 flavors of bottom water in this region, is formed. And so that was very exciting as well. Sorry, I'll just go quickly through this. I realize I'm running out of time. So this is adding to this time series of contraction of bottom water along I-9. So this is I-9 section all the way from the southern end to the northern end. And you can see from the green to the orange to the red to the purple that that bottom water layer, as defined by a particular isopignal, is contracting, so is sinking. And that is even occurring on the northern side of the Southeast Indian Ridge, so in the basin to the north as well, that South Australian basin. You can see, again, in different colors, that the red line is the shallowest of all of those, so that again, the top of that bottom water is depressing. If we look at in TS space, so in temperature salinity space, this is along 150. This is all preliminary data that was calibrated on board, but is not the final version of the data set, calibrated to within GOSHIP standards on board though. So these are good data. You can see that from the 90s through to 2010, 2012, where the Ross Sea bottom water now is just freshening, continuously freshening, had a large rebound in salinity in 2018, and it is starting to freshen again. So we can see that interannual variability playing out in these properties. And as a function of latitude, it looks like this. So you have fresher Ross Sea bottom water than what was found in 2018. So that red compared to that lighter orange. So you can see a continuation of the long-term freshening trend, excuse me, with a small salinification in the mid 2010s, and then it's continuing to freshen again. This is looking at all 5 of those short Antarctic margin sections, and you can see that there's a lot to pick apart here. And so we have kind of our work cut out for us. But one thing that immediately jumps out is that 130 looks very different this year, the red dots, than it did all the other years. And this is the salinity there is very fresh compared to other years, and looks a lot more like the bottom water properties that are usually found at 140. So that's something that definitely jumps out at us immediately. And you can see that 130 transect is fresh, and it's also very high in oxygen compared to previous occupations, suggesting that it's a newer bottom water, so it's been on the shelf more recently, perhaps, than other years. And then we were in the Adélie Depression, and we found that it's cold compared to most recent years, so those orange dots, and higher in oxygen. So again, just preliminary data and no real inferences and implications that Anvita was having. It's more of just a show and tell kind of situation here. And so this is the last slide. We take an integrated and multidisciplinary approach to understand the Southern Ocean's influence on Earth's climate. It was a very ambitious, we kept being told, and highly successful, is what we say, research voyage that occurred earlier this year. So, of course, a big thanks to the captain and crew, the support staff, and everyone who helped make it possible, both on shore and on the ship. And we see some some changes in Antarctic bottom water, some that are expected, like the continuing contraction, and maybe some that were not so expected, like the unusually fresh and high oxygen water at 130. And so that is all I have. I hope I didn't go too far over time.
Not at all.
Okay. Does
Thanks,
anyone
Annie.
does anyone have any questions?
So it's hard for me to see hands, so please jump in either in the chat or just go for it. Because I can't see everyone all at once. I have a comment,
Okay.
which is which is that it's
That's
wonderful
easy
to
for me.
see. It's wonderful to see the data, even if it's preliminary. And I can think of lots of things that we could dig into potentially with some of the biogeochemistry data from repeated stations really far south too. So very exciting to see it. And I would echo that it was definitely a ton of work, and say thanks back to you on behalf of those of us who did not get on the ship for 60 days, and a big success. Sarat, I see you have your hand up.
Thanks, Annie, for your nice presentation. It's interesting to know how the ABW is impacting the overturning circulation, or is going to impact. But my question is slightly different. Now, when you do on-deck incubation, do you maintain the temperature, or how it happens? Like, for example, you take sample from subsurface. Do you maintain temperature, or
So the deckboard incubations were taken using surface waters for the most part. And the large mesocosm experiments on the front deck, we did not maintain the temperatures. In fact, they got quite warm at some point. And the smaller incubation experiments, again, I do not think that we are able to maintain the temperatures, but I can't say for sure, so don't quote me on that.
Thank you.
I don't see hands, but if someone has a question, please just.
Yeah, I think Kohei has one question. Yeah,
Oh.
Yeah, go ahead.
Oh, my video doesn't work, but anyway, yeah, I'm very interested in about the colder and higher oxygen shelf waters. Is there any report saying that some anomalous high dense shelf water production or CS production around there?
Yeah, that's a great question. I haven't looked into it yet, in terms of the sea ice state at the time or in the past couple years. But that's definitely something that we're interested to unwrap.
Yeah, I'm very interested in these things. So thank you.
Thank you.
Any other questions for Annie? No. All right. Well, then I will just take a last opportunity to thank both of our speakers and also everybody who attended and spread the word. I see that Kylie was trying to put some of that stuff into the chat. So we are aspiring to have more frequent webinars as part of the Indian Ocean Sector Regional Working Group. And there's also a newsletter where we aspire, say we, it's mostly Kali, to get publicity of people's work out to the general community. So if you have papers that you have published or that your students or colleagues have published that you think would be of interest to the broader community, please let us know so that we can promote them in our newsletter. And if you would like to volunteer to give a webinar, or you'd like to volunteer a student or a colleague, please also do that. And you can get in touch with us via the SOOS website. We're trying to build a more collaborative community. It's a large region, it's often difficult, but I think this is a great first start. So thank you very much to everyone for being here, and to Kahi in particular for helping us to organize it.